Hi friends, welcome to this video session. In today's video session, we are going to study about or discuss about the DNA replication in eukaryotes. So basically, before diving into the topic, what is the major difference between eukaryotes and the prokaryotes? Why the replication is different in eukaryotes as well as in prokaryotes? We'll deal about about the introduction part. Then we will go about the initiation process in this video. So the entire DNA replication can be divided into three topics or three parts. Initiation, then is the elongation, then is the termination. So I made it into three different videos so that each of the section can be discussed in detail. And also the time constraint won't be much elongated if each is discussed individually. So please do refer to the other two parts also for a better understanding of the complete concept. So now let us go into the topic DNA replication in eukaryotes. So let us know what is eukaryotes in the first part. Eu in the sense, eu means true. Eu means true. Karyon means nucleus. So eukaryotes are those animals that have well developed true nucleus which is covered by a true nuclear membrane. So what is the difference between prokaryotes and eukaryotes? So pro means primitive, karyon means nucleus. So they have a primitive nucleus and the nucleus is not very well developed and they are not covered by nuclear envelope. Whereas in eukaryotes, they have very well developed cell organelles and all the organelles including the nucleus is covered by nuclear envelope. So there is a major difference. And here if you see, the DNA is associated with proteins called as histones and other octamers, whereas in the prokaryotes the DNA is naked. DNA is not associated with any proteins and DNA is almost circular and simple, whereas here the DNA is not circular, it is elongated and much complexity is evolved over here. So that is the reason why the DNA replication also varies. But if you see the major of the process, the enzymes involved, the mechanism is almost similar. Only here and there are some extra enzymes being added and the replication processes varies. So let us start now. So I hope you are clear with what is eukaryotes and prokaryotes. Now let us know the meaning of replication. What is the meaning of replication? So if you see the DNA, as we all know, DNA is a double-stranded helix structure. We consider this as the parental DNA. This parental DNA undergoes replication to give rise to two daughter DNA, like similar to the cell. How a single cell undergoes division or fission or fusion to form two daughter cell. Similarly, single DNA undergoes replication to form a two daughter strands of DNA. So this is one strand and this is another strand. Similarly, each of the daughter strand undergoes again replication to form another DNA. So this process goes on. Again, each daughter cell undergoes to form another DNA strand. Again, each undergoes to form another daughter strand. So like this, the process continues. Each and every daughter cell so formed undergoes replication to form many copies of DNA. So this process is called as DNA replication or duplication. So DNA replication is a process where a single parental strand of DNA produces or gives rise to multiple copies of DNA containing the same genetic information as this parental strand. Whatever the information is there in this DNA, same information will be there in all the multiple copies so formed. So this process, this biologically enzymatically catalyzed process, so it occurs in all biological organisms, hence the name biological processes. And this without enzyme catalysis, this process cannot occur. Hence it is called as enzymatically catalyzed process or enzymatically driven process. So DNA replication or DNA duplication is a biologically enzymatically dependent process where a single parental strand produces multiple copies of DNA containing the same genetic information. It is the exact copy of the parental strand. So this is the meaning of the word replication or the DNA replication or duplication. So now this is the main thing, this is the main aim. We are going to see how a single strand is going to give rise to two DNA daughter strands. How it is going to do. That is what we are going to study in this session. Now, how is this DNA associated or how is this DNA formed? We all know that if you consider a plant cell or animal cell, it has various cell organelles like mitochondria, endoplasmic reticulum, Golgi complex, lysosomes. There are so many cell organelles involved. One of the main major master organelle is your nucleus. This nucleus has nucleolus and it has a nuclear envelope, which will be this is the structure of the nucleus, and inside this nucleus is the chromosomes located. So depending upon the type of organism, the number of chromosome varies. If it is humans, there are 23 pairs of chromosome. If it is onions, there are 6 pairs of chromosome. So depending upon the various living organism, the number of chromosome also varies. But if you see a single structure of the chromosome, if you take a single structure of the chromosome, it has a central region called as centromere. So it has a central region called as centromere, and these are the chromatids, arms of the chromosome we call it as. So to this centromere are attached this arms of the chromosome or chromatids. What is this chromatids or what is the arm of this made up of? It is made up of DNA. So they are minute thread-like, filament-like structures called as DNA. It shortens, thickens, condense to form this chromosome. So whatever I am drawing now, it is nothing but your DNA. So there are filaments or thread-like structure called as DNA. This DNA, so this double-stranded DNA, it shortens, it condenses, it thickens. Because of its large size of the DNA cannot exist as such in the chromosome or in the nucleus. So that's the reason why this long DNA, it thickens, it condenses, it shortens to form this chromosome structure. So if you take the chromosome, it is completely made up of DNA, nothing else. And if you see, this is the DNA strand that I showed you. Each DNA strand is associated with certain amount of proteins. So this is your proteins. It is associated with proteins. This is called as your octamer. So everywhere you can see the protein association. This proteins is called as your octamer. Octa means 8. So there are 8 subunit of histone proteins. 8 subunit of histone protein. 8 histone protein comes together to form a unit known as octamer, and this octamer is coiled by the DNA. So how is it coiled? I'll just show you the pictorial representation. So if this is your 8 subunits, 4 subunits are this side, the 4 subunits are on the other side. 3D, imagine 3D structure, and this is your DNA strand. So it coils like this. So this DNA strand coils the 8 subunits of octamer. So if you see in the electron microscopy, you will find like this region. So if this is your DNA, if this is your double-stranded DNA, and here and there are your octamer units, this DNA is coiled. So it looks like bead on a string arrangement, like the beads, how the beads are arranged on the string. This DNA is considered as string and the green dots are considered as beads. That is nothing but your octamer protein units. So it looks like bead on a string arrangement. This structure is known as nucleosome. So this is just the basics. So this is called as packing of DNA. We call it as packing of DNA helix. DNA is packed in such a way. So this is called as nucleosome or bead on a string arrangement. So if you take this, each of the DNA, if you zoom and see, it is made up of double-stranded structure. So this is how the DNA is packed in the chromosome. Now, as I said you, this DNA is very long. They say that the entire DNA of a single cell of human cell can cover the entire earth 6 times, or it can reach from earth to the sun so many times, they say that. So it is such long this DNA is. So if you see, this DNA is very long, as I said you, right? So it will have, so let us consider this is your chromosome inside which the DNA is present. I have just pulled out one thread of DNA. So this is a double-stranded DNA pulled out. So imagine if the replication is starting from here. Replication is nothing but the copying, not duplication process, as I said you right. If the replication is starting from here, by the time the replication goes on to the entire strand and it fills the by the time the replication for the entire chromosome to be covered, it requires very long period of time. So that's the reason replication starts at many places. That is one of the major difference between the prokaryotes and eukaryotes replication. Whereas in the prokaryotic DNA replication, the replication starts only at a single point. That point is called as ORI site, origin of replication site. But here, the replication starts at many places. Let us consider the replication starts here, and the replication starts here, the replication starts here. So, from many places at single time, the replication starts. So, this place where the replication starts is called as your ORI site, origin of replication site. This is the process, this is the point where the replication starts. So this can be asked again for 2 marker if any examinations you are taking. So what is the ORI site? ORI site is a particular point in the DNA where the replication is initiated or the replication starts or the replication begins. So that point is called as your ORI site. Site is called as ORI site, which I said it is called as origin of replication site. What makes that site specifically called as origin of replication is the sequences in them. So they have a special sequences known as autonomously replicating sequences, ARS sequences. So they are called as autonomously replicating. Autonomously in the sense independently, they are not dependent on any other enzymes or factors or proteins. They can replicate on themselves. Such sequences are present here, which are mainly made up of A and T bases. So it is mainly made up of majorly consisting of adenine and thymine nucleotidic bases are there in this thing. The reason why there is more of A and T rather than G and C is because of the chemical composition of the bond. A and T has only double bond, whereas G and C has triple bond. So double bonds are easier to break when compared to triple bonds. Triple bond, G and C is made up of triple bonds. They require lots of energy to break. So the A and T are present over here. That's the reason. And these sequences are called as autonomously replicating sequences. By estimation, experimentally, they have proved that Saccharomyces cerevisiae yeast has 400 such sites. 400 such sites. Yeast is such a small unicellular, we can say it as microscopic unicellular organism. Such a small organism itself has 400 origin of replication sites in its genome. So you can consider, you can imagine the amount of ORI sites in human DNA. So they've estimated that there are 40,000 to 80,000 such ORI sites in case of your human DNA. So in all such sites, the replication will start at a time and it will happen. Consequently, it will happen. So this is about the ORI site. So this site is very, very important when dealing about the initiation processes. Next is, where does this replication occur? In which phase of the cell cycle does it occur? So as we all know, let it be any living organism, the life of the living organism can be called it as life cycle. So for example, if you take any flowering plant, it can be called as a vegetative growth, reproductive phase, senescence age. So it is a life cycle of a plant. The vegetative or the sprouting stage, the vegetative growth phase, then is the reproductive phase, then senescence or death. If you take of any living organism like human beings, birth, childhood, infancy, childhood, and you can call it as teenage, adolescent age, adult, and elders or senior citizens. Similarly, the cell also has called as cell cycle. So cell has its own phases called as cell cycle. So first is the M phase, next is G1 phase, S phase, and G2 phase. M phase is called as a mitotic phase or the meiotic phase, where the mitosis and meiosis occurs. For example, let us consider this as an adult cell. This adult cell undergoes mitosis or meiosis. If it undergoes meiosis, 4 daughter cells will be formed. If it undergoes mitosis, only 2 daughter cells will be formed. So this phase is called as your M phase, which is also called as mitotic phase or a meiotic phase. So now the daughter cell has been given rise to from this parental cell. Each daughter cell will undergo growth now. It will accumulate all the food materials and it will grow in size, and it will take part in various metabolic and physiological functions. This is called as G1 phase, or the growth phase, or the gap phase, we call it as. So the formed daughter cells that is formed from the meiotic phase or mitotic phase, now it undergoes growth phase. It gets nutrition and it undergoes in various metabolic processes. It takes part takes part in respiration, reproduction, if it is in case of plant, photosynthesis. So that phase is called as G1 phase. Now each of the daughter cell will undergo DNA replication or DNA synthesis. Inside this DNA is present, right? This DNA will undergo duplication. Now this DNA will undergo duplication, that is nothing but replication. That is called as a S phase or a synthesis phase. Synthesis, the name itself says, it synthesizes new DNA. So it is called a synthesis phase. Next we have G2 phase or gap phase or growth 2 phase. Here what happens is, now the cell accumulates all the proteins, enzymes, nutrition required for the mitosis to occur. Again this will undergo mitosis, giving rise to 2 more cells. Right. So for this mitosis process to occur, it will accumulate all the proteins, nutrition, enzymes that is required. Right. So this is called as G2 phase. So this entire is called a cell cycle. M phase, G1 phase, S phase and G2 phase, where in the S phase of the cell cycle, your DNA replication process occurs. But also experiments have proved that the replication gets initiated in the G1 phase. It does not wait until S phase to come. All the enzyme, all the complex formation, all the proteins that are required, all that initiation occurs in the G1 phase. But after the initiation, the replication does not start. It waits for the signal. When the cell enters into the S phase, only the replication starts. So only the initiation process starts over here. So that is about your cell cycle. What is that initiation process? So as I mentioned earlier, the DNA replication can be divided into 3 major phases. That is your initiation, elongation and termination. In this video, we are going to study only about the initiation. So initiation, what happens under initiation is 4 major things happens under initiation. Only this is I'm dealing only with the eukaryotes. So first thing is the formation of pre-replicative complex. Formation of pre-replicative complex. Second one is the formation and activation. So first is the formation of pre-replicative complex. So the second one is the activation of this replicative complex. Pre-RC means pre-replicative complex. Next, third one is unwinding. Fourth process is priming. So these 4 things happen in this process. So let us study each one by one. Pre-replicative complex. The name itself says pre. Prior to replication, a complex is formed that is called as a pre-replicative complex. This complex is made up of by the joining or the accumulation of n number of proteins and enzymes, out of which only the important proteins and the important structures I am going to show you now. So looking at this diagram here, as I mentioned you, this is a double-stranded DNA structure. There are certain sites called as ORI sites or origin of replication site. This origin of replication site is identified by a complex, hexameric complex called as origin recognition complex. That is the first step that is happening over here. So what happens here? This is your double-stranded DNA. It has certain sites called as ORI sites, origin of replication site. This site is identified by a complex called as origin recognition complex. ORC, we call it as in short form. O stands for origin, R stands for recognition, C stands for complex. So it recognizes that ORI site. It is a hexamer, I said, right? So it is made up of 6 subunits. We name it as ORC1, ORC2, ORC3, ORC4, ORC5, ORC6. Each of the subunit is coded by a separate gene. ORC1 is coded by separate gene, ORC2 is coded by separate gene, ORC3 is coded by separate gene. So there are 6 subunits, it is coded by 6 genes. Now what happens? This is the first step that is happening. So ORC, the entire complex comes and recognizes your origin of site and it binds over there. After this, 2 important proteins called as CDC6 and and CDT1 comes and joins. CDT1, CDC6. So now if there are 1000 origin ORI sites, in all the 1000 ORI sites, the process will occur simultaneously. So this is the first part that happens is the ORC. For this ORC, origin recognition complex comes and binds to the ORI site. To that ORI site, after binding this complex, two important proteins called as C6 protein, CDT1 protein comes and joins. As I said you, how much ever ORI sites are there, all sites simultaneously this process will occur. CDC6, the full form of is CDC, cell division cycle 6 protein. CDT is chromatin licensing and DNA replication factor 1 protein. So that is the full form of all this protein. Next what happens is CDT1, CDC6 protein comes and binds over here. CDC6 cell division cycle 6, CDT1 chromatin licensing and DNA replication factor 1. These two protein comes and binds to this. This as soon as this protein comes and binds, it signals the mini chromosome maintenance protein, MCM we call it as. This MCM, mini chromosome maintenance protein, comes and binds to this origin recognition complex. So this is the next step that happens. This mini chromosome maintenance protein is also again an hexamer. It is made up of 6 units, 6 subunits I can call it as. So it is also MCM2, MCM3, 4, 5, 6, 7. So MCM2 to 7. Mini chromosome maintenance. The name itself says these proteins can maintain small amount of DNA. Mini chromosome. Mini chromosome in the sense small. Mini, it's small. Chromosome is nothing but DNA. So it can maintain small amounts of DNA. It is very much necessary for the elongation, initiation of the DNA replication. Without this, DNA cannot replicate itself. So when this mini chromosome replication protein comes and binds here, there is instability causing. So to remove that instability, these two proteins are very much necessary. And also wherever these proteins are there, it acts like a marker molecule. There only this MCM comes and binds. This MCM can't go and bind anywhere else. It cannot bind here. It can bind only to the origin of replication site where the origin recognition complex is formed, and it is marketed by this two protein molecule. So this protein molecule function is maintaining the stability and also in helping the MCM to bind to this complex. Once this MCM has binded to this complex, this protein gets denatured or degraded. Its function is over. Once this MCM comes and binds over here, its function is over, it gets degraded. Next, another enzyme called as CDC45 comes and binds. CDC45 and two more kinases called as CDK and DDK. Cyclin dependent kinase, DDB4 dependent kinase. The full form is, you can Google the full form. And so these are two kinases enzyme, and then a protein called as CDC45 comes and attaches over here. I'll show you the function of these two kinases. Kinases, they phosphorylate this MCM. Phosphorylate in the sense they add phosphate group. They add phosphate group to this MCM protein or to this complex. Now this entire thing is called as your pre-replicative complex. So what is pre-replicative complex? Where your ORC is bound to the origin ORI site, CDK protein, CDC protein, CDT protein, and kinases, CDK, DDK kinases, and this mini chromosome maintains. So how many proteins and how many enzymes are bound to this site, forming a complex called as a pre-replicative complex. This complex has to be activated or it has to be triggered. So that is done by the phosphorylation property. Only if the phosphorylation occurs, the replication will start. That phosphorylation is done by these two kinases called as CDK and DDK. So did you get this? Then what is the function of this enzyme CDC45? This CDC45 protein, it accumulates or it signals all the enzymes required for the replication to get attached to this ORI site. We need DNA ligase, DNA polymerase, we need topoisomerase or gyrase, and we require RNA primers to be synthesized. So many things we need. We need helicase enzyme. So all those enzymes, it signals all those enzymes to be located over here or to come here and settle down. So that is the function of this CDC45. Now, very important thing you have to know is, along with this, you also have another protein called as geminin and genes. So along with this also is very important. Geminin and genes. This geminin is attached to CDT1 protein. As long as this geminin is attached to the CDT1, the CDT1 cannot function. If CDT1 cannot function, DNA replication won't occur. So this geminin has to be degraded or it has to be denatured. This degradation or denaturing will occur and the CDT1 will be activated and replication will start only when the cell enters into the S phase. As long as cell is in the G phase, this geminin will be attached to CDT1. And as soon as the cell enters into the S phase, geminin gets denatured, CDT1 gets activated, replication occurs. So there is also, it is very important to know that if geminin is present, replication won't occur. It is like barrier for the replication. So using this concept, many researchers or scientists have applied this on the cancer cell. So they made such a way that they, I can say they have genetically engineered those cancerous cells in such a way that geminin is always attached to the CDT1. It does not get denatured, so the replication won't occur. So the cancer cells won't multiply. But they have failed in the experiment. They have come to conclusion that if the geminin is always binding to the CDT1 in the cancer cell undergoes replication through another process. They won't follow this process that I said you, right? Pre-replicating complex activation. They won't undergo this processes. They undergo the DNA replication through another pathway. So they come to this conclusion. So the geminin does not play such important role in case of cancerous cell. So this is a new research thing. So I thought let me include. Now one more thing is a genes. Genes is also made up of 4 subunits. These 4 subunits are called as SLD5, SLD5, PSF, PSF, 1, 2, 3. So it is made up of 4 subunits. In Japanese language, 5 means go. The name for 5 is go in Japanese language. The name for 1 is ishi. Name for 2 is ni. And name for 3 is san in Japanese language. So if you take the first letter G-I-N-S, you get acronym called as genes. This genes also it is responsible for the initiation processes, activation and elongation processes for this DNA replication. So this is all about the first 2 parts. Formation of pre-replicative complex, activation through phosphorylation. Next what happens is your unwinding process occurs. Now this double-stranded DNA, once all the proteins or all the complex gets accumulated over here, this complex acts as an helicase enzyme and it unwinds. So now it unwinds to form a fork-like region. So now the double-stranded like this, it gets unwinding like this, and this is called as forking. This region is called as forking. So if you consider the entire strands of DNA, there are many forking sites. This is called as a replication fork, replication bubble or a replication fork. This is a forking site, the ORI site, where this complex is present over here. 3 replicating complex is present. This is the third step, unwinding. So this, how does this unwinding occur? By the process or by the enzymatic activity of helicase. The last step is your RNA primer formation. So remember this, very important. DNA replication won't occur as long as the RNA primer is formed. What is RNA primer? RNA primer is nothing but the short nucleotides of RNA. So on the short nucleotides of RNA is formed. Short nucleotides of RNA called as RNA primer. short nucleotides of RNA called as RNA primer is formed. This event or this process is called as priming, and it is done by an enzyme called as DNA polymerase. It is an enzyme called as a DNA polymerase. So thus the replication gets initiated. So much is about the initiation. So the DNA replication can be divided into three phases: initiation, elongation, termination. Under initiation, it can be further divided into formation of pre-replicating complexes, where all the complex molecular structures formed with the help of the proteins, ORC complex, many chromosome maintenance proteins, CDC45, CDC6, CDT1, genes, then geminins. So you have to write about all this. It's asking in examination. Then is the activation. Activation is done by the phosphorylation of the two kinases, CDK and DDK, cyclin dependent kinase, DDB42 dependent kinase enzyme. It gets phosphorylated and gets activated. Next is the unwinding. So it gets unwinding to form replication fork by the helicase activity. Then last is the priming. Priming is for the short nucleotides of RNA called as RNA primers have been added by the enzyme DNA polymerases. So this is now the replication is initiated. Now the replication is going to start. So let us see what happens in the elongation and termination. How does it get terminated in the further videos? Please do watch it. Thank you. Do like and subscribe.